Yo, and welcome to Shooting the Soil. Today we're looking at an indoor garden bed. It's a 6x3 bed. We just harvested our first run in this bed and starting on our second. So we are taking a look to see how the biology fared and if we're still going strong down there. So far we got some nice cover crop coming up in the bed. And let's see how it looks. Looks like we got a little hyphae right there in the middle. Right now we are at 100x using the 10x objective and just starting our scan around. Nice big organic matter there. Some good aggregates. Yeah, seeing a little bit of fungi. I want to zoom in on whatever this is right here. It looks like a snail out at 100x, but I think that might be a fungal hyphae sprouting. So let's zoom into 400 here and give it a closer look. Gonna need, oh no, more light. And what do we got? Aha. It really does look like a snail, but let's see if we can get a little bit better focus, maybe a little bit of shading. Yeah, right about there maybe. A little more light. Yeah, I think that's we got a sprouting spore of some sort. It's funny, it does look like a little snail with a shell and two little antennas, but yeah. I think that's a new fungal hyphae just sprouting. It's a good sign. Oh, and we got a fungal spore there, another one there, and now down here at 400x, we can see. Oh, we just saw something move up here, but we can see all the bacteria. And look, I think we got a flagellate. Yeah, I've been wanting to see more of them. Is he bumbling or is he going straight? Is it a little ciliate? That's a good way to tell. The ciliates will tend to swim very straight, and the flagellates, since they only have one whip flagella, will tend to bounce around more off stuff. So this one's looks, I would say it's more flat flagellate. It's not very round. It's kind of bumping off stuff and changing direction. So I would say that's a flagellate, part of the protozoa family. Oh, there it is. Out the other side. Swimming around doing his thing. Alright, let's see what else we see here. Yeah, it's just a little salt crystal in there. Thought it might be an amoeba, but nope. Just a chunk of stuff. And we got a little piece of a fungal hyphae there. So hoping to see a little bit more hyphae in there. We added about 2 weeks ago, well maybe like 3 weeks ago now. We did some rootwise. Added in some microbe complete. This is a very strange. That almost looks like a diatom. It's too straight to be a fungal hyphae, but more diversity the better. That's interesting to see, but another nice spore. Let's look at this. That looks more like a hyphae. Oh, I think we got two of them. This one sort of looks a little torn over here, but here's another clearer fungal hyphae. See if we can see any separations. Kind of thought I saw one right there, but yeah, not too shabby. We see a nice cell separation in this little piece of fungal hyphae. That's what we want to see. Oh, well, yep, here we go. More hyphae. Nice little segment there. See where this goes over here. Clearer hyphae, but still good. As long as they're not overwhelming the whole sample. You want that nice diversity. This is interesting looking. A couple clumps of, almost looks like a small little piece of rhizobium or something, or maybe just some yeast cells. Alright. A little bit more focusing. See what else we can find. A clear strand. Probably a saprophyte. Good decomposers. But then we see all these little chunks of fungi breaking down and holding those aggregates, forming that network to bind and hold your nutrients in the soil and provide all the good functions. What do we got here? Not moving. Looks like a nematode tail, but I don't know. There's no stuff inside of them. Doesn't look like there's a head left, but oh, another little flagellate bumbling around in there. Where did that one go? Yep. Yep, not moving straight, bumping off everything. Another flagellate. They're good to see. Nice facultative anaerobe. And we're not seeing a bunch of big ciliates, so we know we're not pushing the line of going anaerobic. We got a good amount of oxygen in the soil to create a nice, diverse community of microbes. Yeah, we got another clear one, probably a saprophyte, commonly found in compost, and they are good decomposers. Alright, what else we got floating around in here? Got some more spores. Another nice round spore. And a good amount of bacteria. Let's see if maybe things are looking a little fuzzy. Oops. That's the dark field filter. See if we can get just things. Yeah, it's looking a little bit better. Now we can really see the different shapes of the bacteria. We got a rod one there, pill-shaped one there, some round cocci. Looking like some pretty good. You want to see just a bunch of different shapes and sizes. Get those lactose bacillus in there doing their thing, along with everybody else. And yeah, they'll be doing their thing right at the root tip, performing rhizophagy, getting sucked inside the root, and the plant can actually digest them. And some are nitrogen fixers, which as they lose their cell wall, they'll release nitrogen directly into the plant root as they rebuild their cell wall to survive and live in the super oxidated environment, which is in the root. So we got an amoeba right here. Another good sign of a healthy soil food web. And more little fungi pieces. But yeah, decent amount of fungi. I would like to see a little bit more. Really want to get certain that I'm to that 1-to-1 fungal to bacterial goal. So I still think we need a little bit more hyphae, but we're almost seeing a nice piece in every frame, but still want to see a little bit more. Another spore there. So we got the spores, so if they pop, we should get some more hyphae. And now we got some living plants growing in the second run in this soil bed. They'll be feeding the soil and promoting all of our microorganisms. That's the whole regenerative idea, to make the soil better and better. Keep that whole ecosystem functioning. So yeah, here we go. So this is a nice example of a brand new fungal hyphae. This looks like the spore, and we are sprouting. That's a really dark one. Go fungi. Yeah, we're getting busy up on this section of the slide. A lot of nice aggregates. Yep, and here we might have, could be plant matter, could be plastic. This is where I really wish I had the epifluorescence. Someday we'll get one of those microscopes, because it's really easy to identify microplastics, because they'll glow if you hit them with epifluorescence. Alright, let's see what we got over here. Some more fungal strands. Looks like we got a nice brown one mixed in with some saprophytes. But good healthy, still seeing a good healthy ratio. Alright. Take a quick look at this nice big old aggregate with all those nice golden brown humus color to it, or humic color, from all the bacteria releasing their acids and compounds. It's either a cysted organism or could be a very round amoeba, but it's probably something in a cysted organism there. Also could be a large spore. It's very round. Some spores are clear like that. More of the saprophyte spores, but that's one of those. But yeah, main thing is seeing that nice diversity. We got some funky, weird looking strands here. They're not uniform. That's why I say they're funky. All kind of twisted up there, but still looks just like a saprophytic fungal strand. I don't think it's actinobacteria or anything. Oh. Alright, let's see what else we can find. Another little fungal. Nice big chunk there of fungal hyphae. And forgetting what we've looked at so far, but I don't think we've been up here towards the top of the slide quite yet. There's some cool looking aggregates. Yeah, good healthy amount of bacteria. Yep. Focus in there. Yeah, nice. Looking nice and aggregated. Pretty healthy. Work on getting a little bit more fungi in there. Yeah, all in all, it's looking pretty healthy. Alright, let's see, maybe we'll we're talking about the dark field, and we haven't done any dark field in a while, so let's throw the dark field filter in. So the light just is now going to hit the slide from the sides, and let's see, we gotta get a little bit more light in here. Oh yeah, we gotta take off our shading. Make sure we're right up towards the top. Alright, let's see if I can get this dialed in here. Maybe a little bit more ISO. That should be good. Nope, less speed. There we go. Starting to see some stuff. And right about there. Alright, and now we have the dark field. Just a whole nother way of looking at things. Probably closer to what the it actually looks like to the microorganisms. They're not just getting blasted with light this way. And works pretty good for doing a 100x scan and spotting some critters moving around in there. Let's see what we can find. See if we can find a nematode or maybe a rare ciliate. But yeah, I really haven't seen many large ciliates in this bed at all. But have seen a decent amount of nematodes in the past. Usually about one per slide. Good healthy ratio. And I see how that organic matter really glows in the dark field sometimes. A little piece of a leaf or something breaking down. Yeah, it's always interesting seeing like different soils from different areas. I want to go to some old growth forests and check out that biology. But it's amazing how much the biology in the soil can change from place to place. Even, a couple, handful of yards from one spot to another, you can have all sorts of different stuff living. There's up by the top. Let's scroll back down. Yeah. Hope to see a nematode wiggling around in there, but none yet. Yep, just cruising through space, looking for stuff moving. Yeah, you can see some of these larger hyphae and how the edges glow in the dark field. Here's another. That one's really glowing. Yeah. Poke around in here all day. Oh yeah, here's that round cysted organism that we were talking about right there. Look how the edges of that glow. Or possibly a spore. Yeah. Pretty cool looking. Ooh, look at how bright green that is. Might be phosphorizing because of the phosphorus. And glowing green like that. But might be actually a little piece of phosphorus. Yeah, alright. Looking pretty good. I think we'll probably wrap things up unless anybody has any last minute questions they want to sneak in here. If you're watching later, go ahead and drop any questions in the comments. Be happy to do my best to answer them. Yeah, and just wanted to point out how much that hyphae's glowing. Good stuff. Yeah. Well, glad you guys joined. And gonna be doing a garden tour here. I'm just waiting for a nice day that's not so sunny so we can get some good light for the camera. And gonna bring you through the garden, and we're expanding into a food forest, trying to make our home disc golf course food forest, so we can have plenty of snacks when we're out there playing. Anyway, that's one of the plans for the homestead, and we're expanding, growing more than we did last year. So yeah, hit those buttons if you would be so kind, and then you'll get notified when I post the tour, and we look at more microscopy, and we share our experience as we learn about growing with regenerative soil. Anyway, till next time, we'll catch you guys on the flip side. Happy growing, and happy Memorial Day weekend too. Hope you guys grill up some good grass-fed burgers and other stuff. Maybe some spring onions and stuff like that. Yeah. Way to go, guys.